Secondary growth. Normally, secondary growth takes place in roots and stems of the dicotyledonous plants and the gymnospermic plants. By the activity of lateral meristems, increase in the circumference or girth of the plant organs due to the formation of secondary tissues in stellar and extrastellar regions are called secondary growth. Pteridophytes and monocots have only primary structure, but in gymnosperms and dicots, they soon starts undergoing secondary growth. Here you have to remember, due to the activity of lateral meristem, increase in the circumference or girth of the plant organs by secondary tissues in stellar and extrastellar regions is called secondary growth. Secondary growth generally occurs in roots and stems of dicotyledonous plants and gymnospermic plants. In pteridophytes and monocots, they contains only primary structures. But in case of gymnosperms and dicots, they will start secondary growth by their lateral meristem. Due to lack of cambium in monocotyledonous plants, secondary growth is absent. But exceptionally, secondary growth takes place in some monocotyledonous plants such as palm tree, yucca, dracena, smilax, agave, and coconut plants. In dicots and gymnosperms also, stem and roots only show secondary growth. Leaves does not shows secondary growth in dicots and gymnosperms also. Here you have to remember, the vascular bundles in monocotyledonous plant stem and leaves are conjoined, collateral, closed vascular bundles. Closed vascular bundles in the sense there is no cambium between xylem and phloem. When there is no cambium, there is no secondary growth in that particular plants. Due to that, secondary growth is absent in monocotyledonous plants. But exceptionally, some monocots are showing secondary growth such as palm, yucca, dracena, smilax, agave, and coconut. In dicots and gymnosperms also, stem and root only show secondary growth. In those also, leaves does not shows any secondary growth. Under this we have two topics. The very first one is secondary growth in dicot stem. Second one, secondary growth in dicot root. The very first one, secondary growth in dicot stem. In dicot stem, secondary growth occurred in both stellar region and extrastellar regions. Very first we have to discuss about secondary growth in stellar region. The secondary growth in stellar region begins earlier than extrastellar region. Secondary growth in stellar region or secondary growth in vascular region occurred in following steps. Under these, the very first one is formation of ring of vascular cambium or formation of cambial ring. Cambium which is present inside the vascular bundle is called intravascular cambium. This is one type of primary meristem. First of all, cells of medullary race becomes meristematic to form interfascicular cambium, which is secondary lateral meristem. Intravascular cambium and interfascicular cambium are collectively known as vascular cambium or cambial ring. Vascular cambium is formed in the form of a complete ring which is made up of single layer of cells. In dicot stem, some part of vascular cambium is primary and some part is secondary in origin. Two types of cells are found in a ring of this vascular cambium. One is fusiform initials and the second one is ray initials. Two types of cells are found in the ring of this vascular cambium. One is fusiform initials and the second one is ray initials. Fusiform initials are long with pointed ends while ray initials are spherical or oval. Amount of fusiform initials is more in vascular cambium. The second one, activity of cambial ring. Under this, the very first one, activity of fusiform initials. Continuous periclinical divisions or tangential division takes place in fusiform initials. This type of activity, few cells are formed towards the periphery and these cells differentiated into secondary phloem or bast, and some of the cells are formed towards the central axis and these cells are differentiated into secondary xylem or wood. You have to remember, secondary phloem is also called bast, secondary xylem is also called wood. By the pressure of newly formed secondary phloem, primary phloem and initially formed secondary phloem is pushed towards the outside and get crushed. By the pressure of secondary xylem, all the primary tissues of such primary xylem, pith, etc. and old secondary xylem degenerates in the center of stem. Due to this, central part of the stem becomes woody. These activities are going on continuously in plants throughout the life. Normally, more secondary xylem is formed as compared to secondary phloem. Due to this unequal distribution occurred by hormones, secondary xylem is formed 8 to 10 times more as compared to secondary phloem. Secondary xylem forms in plants regularly and primary tissues degenerates or crushed continuously. The new secondary xylem also degenerate the old secondary xylem. Waste materials are formed in the stem such as lignin, suberin, tannin, resin, gums, etc. Due to degeneration of these cells, all these waste materials are filled in the lumina or cavity of tracheids and vessels of secondary xylem. Because of this, wood in central region of the stem becomes dark colored or black brown. It is called heartwood or duramen. The peripheral or outer wood which looks light in color is known as sapwood or alburnum. As a result of growth of secondary xylem, the diameter of heartwood increases. The primary xylem, however, remains less or more intact in or around the center. Physiologically active wood is sapwood. The main function of sapwood is water conduction or water transportation in plant body. Heartwood is economically important. Heartwood in plants provides maximum mechanical strength to stem. Let's check the diagrammatic representation of formation of cambial ring or formation of the ring of vascular cambium. See, in the first diagram, it is normal dicot stem before secondary growth. When it starts secondary growth, so here a ring-like structure was formed between vascular bundles continuously, which is called intervascular cambium or intervascular cambium or cambial ring. By the activity of this cambial ring only secondary growth was occurred. Key points. If the heartwood is destroyed in any stem, then there will be no effect on plants. Any vital function is not affected. But if the sapwood is destroyed, then the plant will die because conduction of water will be blocked. Study of wood is known as xylotomy. If a wood is exposed freely in air, then decomposition of sapwood takes place rapidly. Position of oldest layer of secondary phloem is just inside the primary phloem. Position of youngest layer of secondary phloem is just outside the vascular cambium. If we remove the hardwood of a stem, it cannot affect that plant. Why? Because it is economically important, but there is no effect on biologically. But sapwood is biologically important. particularly physiologically important in water consumption. If we remove sapwood of a plant, then plant will be die because of conduction of water will be blocked. The study of wood is nothing but xylotomy. The oldest secondary phloem is just inside the primary phloem. The youngest layer of secondary phloem is just outside the vascular cambium. Position of oldest layer of secondary xylem is just outside the primary xylem. Position of youngest layer of secondary xylem is just inside the vascular cambium. If xylem is blocked, then shoot will die first. See, now coming to xylem, the old xylem or secondary xylem is just outside the primary xylem. The position of youngest xylem is just inside the vascular cambium. If xylem is blocked, then shoot will die first. These key points are very important in the exam point of view. Check these points carefully. We already discussed about activity of fusiform initials. Now the activity of ray initials. Periclinical divisions are also going in continuously in ray initials of vascular cambium. Some of the cells are formed towards inside and some of the cells are formed towards outside through these divisions, and these cells are made up of parenchyma. Radial lines of cells of parenchyma are formed in the stem. These are called vascular rays or secondary medullary rays. They conduct water and food in radial direction. Order of development of vascular rays is both centripetal and centrifugal manner. They pass through secondary xylem to secondary phloem in radial manner. Formation of annual rings. Springwood and autumnwood formation. Annual rings are formed due to unequal activity of vascular cambium. The activity of cambium does not remain same. It is changeable throughout the whole year. Activity of vascular cambium is affected by physiological and environmental factors. In winter or autumn season, the activity of the cambium is less and forms fewer xylary elements. The cells formed during this period are small, thick-walled, darker, with higher density and have narrow lumens. This type of wood is called autumn wood or late wood. The vascular cambium is highly active in spring or summer season and produces a large number of xylary elements, and cells of secondary xylem are larger, thin-walled, lighter in color, with a lower density and have wider lumens. This wood is known as spring wood or early wood. The numbers of annual rings formed in a tree give the idea of the age of tree. Study of determination of age of plant called Dendrochronology. The annual rings are counted from the base of the stem because basal part has maximum annual rings, upper part has lower annual rings. Therefore, counting from the basal region can give the correct idea. The width of annual ring is less in unfavorable conditions and more in favorable conditions. Thus, the annual rings are also indicative of environment period the tree has encountered previously. The autumn and spring wood are formed in the form of rings. The rings of any type of wood is called growth ring. Thus, two growth rings are formed in 1 year and they collectively constitute a annual ring. A piece is taken from the stem, from the base of the stem with the help of increment borer instrument. The annual rings are counted from that piece and again inserted or fitted into the same stem at the same place. More distinct annual rings are formed in that region where climatic variations are sharp. Distinct annual rings are not formed in tropical plants. More clear annual rings are formed in deciduous plants as compared to evergreen plants. particularly in temperate region. Least distinct annual rings are formed in seashore regions because climate remains same throughout the year. In deserts, annual rings are less distinct. Distinct annual rings are not formed in India except Himalayan regions. More distinct annual rings are formed in temperate plants. The study of age of a tree is called Dendrochronology. The study of age of plant was done by its annual rings. Annual rings are formed by spring wood and autumn wood in an year. All these points are very important in exam point of view. Check these points carefully.